Okay, so just to give you some background on the 2014 Ebola outbreak. So it was the largest outbreak of Ebola. All previous outbreaks had seen very few numbers of casualties. The largest prior to that was around 400 deaths. But in this outbreak, we saw over 28,000 cases and more than 11,000 deaths. While the disease has largely gone away, there have been multiple flare-ups in 2015 and 2016. This page from the CDC shows the most recent outbreaks, and we can see even in March this year there were 13 cases in Guinea and Liberia. During the outbreak, we've learned a lot about the virus. For example, that it can persist in immune-privileged sites, such as the eye. So there's an example of a Scottish nurse who had multiple flare-ups of the disease because it was being maintained in her eye. There's also evidence of it being present many months after, even over a year, being present in bodily fluids such as breast milk and semen. And this has also led to the possibility of cases of sexual transmission. So although we've learned quite a lot about what can happen in Ebola, there's still a lot to be understood about the biology of the virus. So we took this basic idea that in this outbreak there were many Ebola virus genomes sequenced. So can we use information present within those genomes to identify molecular determinants of Ebola virus pathogenicity? So what is it? What are the amino acids even that make Ebola virus so highly pathogenic? And we use some basic information about there being 5 different Ebola virus species. 4 of them are human pathogenic. So we have Ebola, which is caused the most recent outbreak and most of the outbreaks that we've seen to date. And then there's Sudan, Bundibugo, and Thai forest. And these are all pathogenic in human. We also have a 5th species, Reston, which was identified in Reston, Virginia, in the United States some years ago in an animal house when monkeys that had been imported from the Philippines were dying, and they identified that they were infected with Ebola. But the animal handlers, while they were clearly had been infected, were not ill. So we have a non-human pathogenic species as well. So our basic idea, we have these 2 different groups. If we compare the genomes, we can identify, hopefully, the genomic differences that may account for this difference in human pathogenicity. So we used a fairly, what I would call, traditional bioinformatics approach, starting with sequence analysis, which then led on to more in-depth structural analysis. So central to our sequence-based approach was to identify specificity determining positions in the Ebola virus proteins. So in this example alignment, this position here would be a specificity determining position. So if we have subgroups within our alignment, so you might think of this traditionally would be thought of as, say, different members within an enzyme family. You have a conserved residue that's present as one amino acid in one group and present as a different amino acid in a second group. So these are positions that are differentially conserved, and they've often been shown to be associated with functional sites and protein-protein interaction sites. So we can then use one group as our human pathogenic species and our second group as Reston, and we can compare them to identify these STPs. So before I show you some results, just to give you a brief overview of the Ebola virus. So this is a really lovely image from David Goodsell. You also see that Ebola has 7 genes, a glycoprotein, a matrix protein, VP40, a nuclear protein, 3 structural proteins, VP24, VP30, and VP35, and also a polymerase protein, L. This image nicely shows the structures that are available. So thankfully there are structures available for nearly all of the Ebola virus proteins, with the exception of the polymerase protein L. We can also see that some regions, so these solid rectangles and circles, indicate parts of the protein that haven't been solved. So although we have some structural data, it is far from complete. So by analysing the 196 different genomes that we had, we identified 189 specificity determining positions. Now, here we have a view of the ebolavirus genome, and we can see these red dots indicate STPs. So there were specificity determining positions identified in each of the 7 genes of the virus. And these numbers here indicate how many in each protein. So we proposed that a subset of these 189 STPs should explain the differences in human pathogenicity from the Western compared to the others. So now we wanted to look at this in greater detail. So which of those changes, what is the subset that actually has this effect on pathogenicity? So we moved into mapping them onto the structures. So we extracted the structures that were available. We did a little bit of extra modelling, not very much. And then we mapped the STPs onto the ebolavirus species, on proteins. And as you can see here, we have some nice structures, and we've mapped on the specificity determining positions. So overall, we are only able to map on 47 of the 189 STPs, and this is partly because, for example, L has 66 STPs, and we don't have a structure available for it. So now that we have those 47, we want to analyse those structures. So where in this protein structure are the STPs? Are they likely to alter protein stability? Are they likely to have an effect on protein-protein interactions? And then if they do, can we suggest a role in pathogenicity? So at the end of the structural analysis, we'd identified 8 candidate STPs. We think there is clear evidence that they have some effect upon the protein structure or function of the Ebola virus proteins. There are a few other candidates where we had weaker evidence. So these were 8 STPs were present in 4 different proteins. So half of them are present in the structural protein VP24. We split them into those that were likely to affect stability. For example, they might remove a hydrogen bond, and those that are present in protein-protein interfaces. In VP24, we see that there are 3 that are present in a protein-protein interface, 1 that's likely to affect stability. VP30 and VP35 each have an STP that's present in an interface, and then for VP40, there's an STP that has an effect upon stability, and 1 that has an effect as an interface. So I don't have time to go through all of these in detail, so I'm just going to show you those that I think are most interesting. So in the protein VP40, in its octameric form, it regulates viral transcription by binding to RNA, and we've identified this STP here. So in the pathogenic species, the 4 pathogenic species here, we see that it's a proline, and in the resistant species, it's a threonine. And it's in this region of around 15 residues that are completely conserved. So these black squares indicate that that position is completely conserved across all of the genomes. Now, this shows where P85T occurs. So it's present in the octamer interface, and we can see it in more detail here. So this is it here. It's in a very short 4 amino acid turn around an inch sheet. And our suggestion is that that change to threonine is likely to have an effect upon this short turn and have some effect upon the conformation of the octamer. What's not clear is how this may affect function and how that may be related to pathogenicity. So second, I'm going to focus on VP24. one of the functions of VP24 is that it interferes with interferon signaling so that it can prevent a host immune response. So this is a very simplified diagram. In a standard infection, STAT1 would bind to cariotherin and it would be transported to the nucleus and we would get interferon signaling response. However, when you have an ebolavirus infection, VP24 is known to bind to caryopherins and also possibly to STAT1, and by doing this prevents the transport of STAT1 into the nucleus and therefore prevents the therefore prevents the host immune response. So when you have the ebolavirus infection, you're unable to have the immune response that your body would want. So we had 4 STPs in VP24 that we identified. The first is here in the protein structure. You can see it enlarged here, and this is position threonine 226, which in Western is an alanine, and the result of that change removes this hydrogen bond with D48. And so our suggestion is that this will introduce some, maybe change some flexibility or have an effect upon stability in this region. So what I'm doing is saving the most interesting for last. So thankfully there's a structure available of VP24 bound to the human cariotherin protein. And we can see that there are 3 STPs listed down here that are present in this interface. And so our suggestion is that as this is an important functional interface, these changes may have an effect on the ability of the Western virus VP24 to bind to the cariotherin, and therefore may affect or reduce the ability of the Western virus to prevent the immune response. So we've taken this a bit further and we've used some methods, MCSM and Foldex, to investigate what they predict the effect on stability and affinity would be for these. Now, most of the effects are fairly small, and even when we combine the 3 together, it's predicted to be fairly small as well. Now, more recently, in collaboration with Franco Frattinelli at King's College London, we've been working on molecular dynamics simulations of the complex. So we've taken the Ebola virus complex with human cariotherin, and we've made a model of the Western complex, and this shows the results from cross-correlation analysis, and what we see is red lines indicate movements that are correlated, so we see that there's lots of correlated motion between the Ebola virus VP24 and the cariotherin, so they have very good interaction. And then for the Western complex, we see that while there are still correlated movements, we have a number of anti-correlated movements shown in blue. So these it's suggesting that this may be a less effective interaction partner. However, there's still some way to go on it. This is still a hypothesis, and we're now, with the collaborators at Public Health England, investigating these changes to see if they do have a functional effect. So that, in summary, more or less leads us to the kind of question: how many mutations are required to alter the pathogenicity of an Ebola virus species? So we have identified a fairly small set of STPs that we think have an effect on pathogenicity. So if we add to that information that Western is known to circulate in pigs in both the Philippines and China, then that leads us to the question: could Western become human pathogenic with a few mutations? It's not clear, but maybe it's something that we should be concerned about. So, just a brief summary: we've identified conserved differences between human pathogenic and non-human pathogenic Ebola virus species. We've then used structural analysis to provide greater insight into those STPs. In VP40, we see the change P85T is likely to have a considerable effect upon the optimal structure. And then most importantly, we think these changes in VP24 may alter binding to carrioferin and have an effect on the inhibition of interferon signaling. And then finally, how many of these changes would be needed to make the resting virus human pathogenic? So, just to acknowledge who did the work, so Morena and Miguel were PhD students in the lab. Before we started this project, I didn't know much about viruses, so thankfully Martin Michaelis and Jeremy Rossman in my department did. And then the molecular dynamics work has been done with Franco Fratinelli at King's College. So thank you for your attention.